Okay, the next topic is humoral immunity. Humoral immunity relates to antibodies. Every single time we talk about humoral is antibodies. So we said that there are two main types of cells, lymphocytes T and lymphocytes B. Lymphocytes T are mainly related to the cellular immunity. Lymphocytes B are related to the humoral immunity. The main reason for that is because the lymphocytes B are basically the ones that are going to be responsible for the production of antibodies. The same way that we talk about the cellular immunity, there are different steps. Same thing happens with the lymphocytes B and immunity. So the lymphocytes B are going to phagocytize certain microorganisms, and they're going to display them in the cell membrane. Okay, so we are going to have that in here. So right here, you can see here antigen recognition. So they identify the antigen, okay, or they can actually get the APC cells, and the APC cells or the T helpers are going to provide the antigens to the lymphocyte B that are going to identify that. So then these antigens will get the information from the lymphocyte B in the sense that they will identify them, and they will start displaying them in the cell. So we have the antigens right here. The lymphocyte B is going to phagocytize these antigens, and then is going to process that, and then the epitopes of these antigens are going to be displayed right here in the major histocompatibility complex. Okay, so right here it says immunocompetent B cells exposed to an antigen. Antigen binds B cells with complementary receptors. Okay, so that's what you have right here. Number two, antigen presentation. The cell internalizer, right, phagocytizes that, it goes inside, then the lymphocyte B is going to process that particular antigen, and is going to display the epitope right here in the cell membrane using the major histocompatibility complex. Basically the same concept that you learn with the cellular immunity. Then once this is available in the lymphocyte B cell, the T helper comes over here, and the T helper is going to identify the antigen being displayed by the lymphocyte B. What the T helper is going to do? Well, the same thing that it did with the cellular immunity, is going to release interleukins. What interleukins are going to do are going to stimulate the production of more lymphocytes B. What is that called? Clonal selection. So basically you clone this lymphocyte B so you can have more and more of this, so you can have an amplified response or amplified group of B cells showing this particular type of epitope. So more and more lymphocyte T helper can actually identify them and help them in order to produce more and more lymphocytes B with the purpose of producing or enhance the response. What happens with this lymphocyte B? They're going to go through a process of differentiation. After the process of differentiation, these lymphocyte B will turn into plasma cells, and some of them will go into memory B cells. The job of the lymphocytes B is to turn into plasma cells, and the plasma cells will produce the antibodies. The response that we call it attack. So attack actually does not mean that the lymphocyte B will, that the antibody will destroy the microorganism. Just basically is going to identify the antigen available in the microorganism, or the epitope available in the microorganism, and it will identify and bind to it, so it can actually be destroyed by the lymphocytes T. So let's keep scrolling down. Let's see what we have next after that. So right here we have this part where it says that most of the obviously lymphocyte B are going to turn into plasma cells in order to produce antibodies. So you have in here two pictures, one of them showing you the B cell, and in here you have the plasma cell that is obviously larger, right? It has a whole bunch of Golgi apparatus in here in order to be able to display the epitopes in the cell membrane. Obviously, if you have a whole bunch of Golgi apparatus, because you have a lot of rock in the plasma reticulum. Okay, so we keep scrolling down, and we have in here attack. Again, attack, I said, is not a matter of the antibody destroying the microorganism, but rather binding to it and identify it as a microorganism, because it's going to have the antigen present in that particular microorganism. So right here we have the antibody. Let's stop for 1 second right here. Let's skip this part for 1 second that is showing you the characteristic of the antibodies. And let's take a look at how the antibodies are going to stop the antigens, or how they're going to make them not responsive or not being able to create or initiate an immune response. So there are basically 4 mechanisms that the antibodies are going to use once they are produced by the plasma cells. The first one is called neutralization, because in this case the antibodies are going to neutralize the antigen by covering it. Right? That's why, since it covers it, then obviously the antigen will not be able to provide or to initiate or trigger an immune response. That's why this is called neutralization. Next one is the complement fixation. Complement, remember, it got to do with the complement mechanism that we covered before. So in this case, IgM and IgG are going to attach to the antigen, therefore producing a particular type of molecule that is going to be called, in this case, an antigen-antibody complex. So this is going to modify the way they are going to be arranged, and because of that, then the complement binding sites that you can see here are going to be available for the complement attached to it, and therefore the complement will attach to this new type of molecule that is going to be formed by the presence of the antibody attached to the antigen, therefore initiating the complement mechanism that eventually will finalize with the inflammation, phagocytosis, immune clearance, and cytolysis, which are the results of the complement activation. So again, in this case, complement fixation is the presence of antibodies that are going to attach to the antigens. Therefore, this attachment modifies the characteristic of this molecule, of this complement, of this antigen, therefore making it available for the complement. Once the complement has these areas available, it's able to attach to the antigen and therefore produce the response that it happens with the activation of complement fixation, of the complement mechanism, which is inflammation, phagocytosis, immune clearance, and cytolysis. The next one is agglutination. In this case, agglutination is the ability that antibodies have to attach to multiple antigens. Since they have the ability to attach to multiple antigens, then they are able to put them together, to bind them together, or to stick them together, therefore rendering them inactive. Okay, and this is obviously agglutination is what happens with blood type. You have an example in here where you see the antibody attaching to multiple red blood cells. Okay, and that's agglutination. Why? Because it has, the antibody has multiple areas of attachment. Therefore, it allows it to attach multiple different types of red blood cells right here, for example, therefore producing agglutination. The next one, and last one that we have right here, is going to be precipitation. It's more or less the same mechanism as the agglutination. That's why it says you see similar process in which antibodies link multiple antigens. Okay, and what the only situation with this is that when they link multiple antigens, they form these big antigen antibody complexes. They are so big that they can actually not remain dissolved in the solution, and because of that they precipitate. They fall into different areas of your body, but unfortunately they can be obviously removed by the presence of macrophages, for example. Okay, so we have agglutination and precipitation as two similar processes. Okay, in both cases you have antigens that have multiple binding sites, therefore they can bind to multiple different antigens. The main difference is that in the case of precipitation, when it binds to multiple different ones, they form large antigen-antibody complexes, therefore they cannot remain dissolved in the solution, and because of that they will precipitate. Okay, but this precipitation can be obviously removed by phagocytosis. Okay, so that's how the last step, which is called attack, is actually being presented here by these 4 different steps that the antibodies can take in order to render the antigen not active. So if we scroll down, we have in here, we have 5 types of antibodies. AGA, right here, AGD, AGE, and we have AGG and AGM. AGM, as you can see here, you see 1, 2, 3 multiple different areas for binding. So in the case, AGM plays an important role in the last 2 that I was telling you about, precipitation and agglutination, because it has multiple areas where it can attach to multiple different types of antigens. Okay, IgM represents 10% of the antibodies present in plasma. Okay, and as you can see here, immune response very strong in agglutination and complement fixation, and obviously that is clearly seen here with the presence of all these areas. Next one, we have IgG, also very important, because obviously is one of the antigens that, one of the antibodies that is going to be secreted in the secondary immune response. It has 1, 2, 3 areas that it can be used for attachment, so it's also very important in complement fixation. IgG also is able to cross placenta, therefore able to provide some immunity to the baby. Okay, IgE, E is related to eosinophils. Right here, eosinophils, very important with IgE because this one is actually very commonly found in inflammatory as well as an allergic reactions, as you can see right here. IgD is important in the B cells, okay, because it helps with the activation of the B cells. IgA is very common in tears, saliva, in the areas that we have mucosa, okay, that's why it's also available in the mucus. So these ones are the ones that we are going to have basically available in all these different areas. So let's take a look at the antibodies. If we scroll down, the part that I didn't mention, right here you have the characteristics of the antibodies. It's going to have heavy chain because it's long, light chain because it's short. This area right here is the antigen binding site. This is the area that is going to change, therefore it's going to be called a variable region. This area and this area don't change, therefore they are called constant regions. They are attached to each other right here using disulfide bonds. Okay, if you scroll down, after all this process happens with the antibodies, we are going to get memory. And the memory obviously is very important because you're going to have antibodies that are going to play an important role in it. You have in here primary response. You have the infection for the very first time with an antigen. Right here you can see in day 5 basically you are going to have IgM that happens first and then IgG. This gives you initiate the memory. What happens if you get another infection with the same type of microorganism? In that case, you're going to see here the secondary response right here. There's no delay in the presence of IgG and IgM. It's an immediate response, therefore not allowing the antigen to cause any sort of problems in our body. And as a matter of fact, you can see how high and how strong the response of our body is with the production of antibodies in order to destroy the microorganism. So very important to remember what antibodies you have in primary and secondary response. Remember, the antibodies are going to come from the plasma cells that are going to be derived from the lymphocyte B. Obviously, the memory is going to be given to you by the lymphocyte B that eventually turns into plasma cells. Obviously, you have a lot of lymphocytes B in the germinal centers of the lymph nodes. And remember that the lymphocytes T are the ones that go on mature in the thymus. But remember, lymphocytes T are mainly for the cellular immunity, and lymphocytes B are mainly for the humoral immunity. Right here you have a nice table showing you all the different agents that are going to work in the immunity. So it's just a nice summary that you should pay attention in case that you get some questions about it. And that's about it.